So I titled the presentation today, as Dr. Wiener said, "It's a Small World After All, Transdisciplinary Approaches to Addressing Racial Health Inequities in the Precision Medicine Era." So in 2015, then President Barack Obama, along with then NIH Director Francis Scott Collins, ushered in the precision medicine era of research. And in a State of the Union address that year, President Obama said, "Tonight, I'm launching a new precision medicine initiative to bring us closer to curing diseases like cancer and diabetes, and to give us all access to the personalized information we need to keep ourselves and our families healthier." And it's not that these ideas didn't exist before, but it started to come to the forefront of our conversation, the forefront of our research dollars, that this is what we're going to go after, is precision medicine. Shortly after that, later that year, Dr. Collins wrote this paper in the New England Journal of Medicine, highlighting the hopes of this precision medicine era. And he had really two components that he outlines of what he wanted this to be. One, he said, we're going to focus on cancer research. Why? Because cancer has a high disease burden and increasing incidence as the population ages. And two, cancer specifically have a genomic signatures with some features that are tumor specific and others that are common to multiple cell types. Despite being in this setting, I am not a cancer researcher, and I certainly am not going to pretend to be today in front of actual cancer people. So I will say that a lot of my work really focuses on the second component that Dr. Collins outlines in this paper, where he talks about transdisciplinary research to address a range of diseases. And I take this quote out of this paper where he says that the initiative will encourage and support the next generation of scientists, that was me because I was in grad school at the time, to develop creative new approaches for detecting, measuring, and analyzing a wide range of biomedical information. And I really key in on this second part where he says, including the molecular, genomic, cellular, clinical, behavioral, physiological, and environmental parameters. So this wasn't just supposed to be a genetics problem, right? We're supposed to understand how the environmental factors, our lifestyle factors, our behavioral factors are all intersecting with the genome, and how all of those things together are actually contributing to health. A little bit later, Miyun Corey from CDC, he wrote a paper talking about the idea of precision public health. And he says, could the same technologies that propel precision medicine usher in a parallel era of precision public health beyond the treatment of sick individuals? That if precision medicine is about providing the right treatment to the right patient at the right time, then precision public health can simply be viewed as providing the right intervention to the right population at the right time. And it's not that these two ideas between precision medicine and precision public health are in conflict. Rather, they're just on opposite ends of the same spectrum, of thinking from individual level to population level. And if we're going to do precision medicine well, and I'm going to come back to this graphic a lot in this presentation, we have to really think about all of the layers that affect health, anywhere from individual level genes, thinking about our individual risk behaviors, our stress levels, our engagement with physical activity, our diet, our exercise. We have to think about the neighborhoods in which we live that are influencing those individual behaviors that we make. We have to think about the policies that are influencing the neighborhoods that we live, that influence the decisions that we get to make, that influence our genes. And I think inherent in this idea of us taking this multi-level, multifaceted approach is that we all have to work together in this. And if you ignore me after this slide, it's okay. I know everyone's tired. But get this point is that the whole point of this presentation, I titled it, "It's a Small World After All," to really get to the point that if we're going to do precision medicine, that we have to think about all of these different levels, and we have to work across all of our different fields. We have to get out of the silos of our own expertise and be able to do this work together to treat whole people, because that's what we are. We're whole people. So it just, a little bit of the bridge that I try to bridge together is looking at social determinants of health and genetics. So most of the time you have geneticists over here, they're like, I guess social stuff matters, and you have the social people over here, they're like, yeah, genetics matters, but ooh, genetics. So a lot of the work that I've been trying to do is try to link what we know in terms of social determinants of health with genetics. So how do geneticists think about disease? I think that we think about disease in two broad ways. One is we think about simple or Mendelian diseases. So simple Mendelian diseases, those are diseases that are caused by mutations in one gene. That's something like a sickle cell, like a Tay-Sachs, for example. The other way in which we think about it is in terms of complex diseases. So complex diseases result from the combination of multiple genes working together, influenced by the environment, the physical environment, the social environment, your lifestyle factors. And a lot of my work really focuses on thinking about the complex diseases. Why are complex diseases important? Well, if we look at the leading causes of death in our country, many of them are complex diseases. Many of them are the combination of genetics and environmental factors. So how do we think about the relationship between the environment and genes in health? Well, I think that there's three ways in which geneticists think about how genes and environments go together. One is the long-standing nature versus nurture debate. What component is nature and what component is nurture? And to do that, we do twin studies. So with twin studies, we have monozygotic twins, dizygotic twins. Those are identical twins versus fraternal twins. And we try to understand what component is genetic and what component is environmental. So if you compare monozygotic twins pretty much have the same DNA sequences. Dizygotic, the fraternal twins, they have 50% of their shared DNA sequences. And what do we try to do is we try to tease apart what part is environmental, what part is genetic. The other way in which we do it is the long-standing, is the gene by environment interactions. Gene by environment interactions try to say, given that you have a certain genetic sequence, how does this genetic sequence operate in different environments? So my favorite examples of that is the idea of perfect pitch. I come from a family full of musicians and can't sing a lick, don't know notes. But the idea of perfect pitch is that people believe that you are born with the genes to have perfect pitch, but you have to be musically trained as a child in order to ever achieve that. So you can have the genes, but you have to also have the right environment to have experienced to have perfect pitch. And more recently, what we've been thinking about is this field known as human social genomics. And this is a lot of where all of my work lies. So in the other two, when we think about nature versus nurture, when we think about gene by environment, it's like genes are over here, environments are over here. What happens if you have both? But human social genomics and human social epigenomics gives to this idea that it's that the environment is actually impacting how your genes are functioning. So it's not genes over here, environment over here. It's that we have the environment, and then how our genes are functioning are dependent upon, how our genes are functioning are influenced by that environment specifically. So it's not totally a new idea. So Nancy Krieger, she's a social epidemiologist out of Harvard. She brings up this idea when she presents eco-social theory, and this is back in 1994. And she says, a central focus is on how we literally biologically embody exposures arising from our societal and ecologic context. At issue are socially patterned, exposure-induced pathogenic pathways mediated by physiology, behavior, and gene expression that affect the development, growth, regulation, and death of our body's biological systems, organs, and cells, culminating in disease, disability, and death. So trying to get to this idea that we actually need to think about how the environment, the how of how the environment is actually affecting how our genes are functioning. Along with that idea is when human social genomics actually started being written about in about 2007, George Slavich puts out this quote, he's from UCLA, writes that an emerging field of research is demonstrating that our external social conditions, and especially our subjective perceptions of those conditions, can influence our most basic internal biological processes, namely the expression of our genes. So one of the early studies in human social genomics was a study by Steve Cole out of UCLA, and he was studying the idea of loneliness. So he had people who filled out a survey that said that they were lonely in their survey and looked across the genome to try to see, is there differential expression patterns by people who said they were lonely versus people who said that they weren't lonely. And he considered loneliness to be a 30% difference in gene expression. So he finds that there's 144 genes that differed in expression just by whether people who filled out the survey said that they were lonely or not. So he says, okay, well, what do these 144 genes mean? And when he did the bioinformatic analysis on it, he realized that there was a difference in immune system-related genes that were connected to a person's loneliness. And this is a survey of loneliness. So it was really groundbreaking, really exciting research. What do we do as researchers when a researcher does something really exciting? We try to do it with our own favorite exposure of interest. So that's what happened. So then people did it on grief. People looked at socioeconomic status. People looked at caregiving stress. And what happened is they found a conserved transcriptional response to adversity. They found the same pattern looking across all of these different exposures, categorized by an upregulation of pro-inflammatory genes, a downregulation of type 1 interferon genes, and a downregulation of antibody synthesis genes. So overall, looking across these different exposures, they're finding changes in our immune system due to adverse social exposures. Why do we think we're finding the immune system? Well, it's because one is just practical. It's a lot easier to get blood samples to do your surveys than like brain tissue. So you're doing the studies on white blood cells, so it makes sense that you're going to see something in white blood cells. But two, it's also, again, the activation of our body's fight or flight response. So your body sees like the stressor, like the lion, like the bumblebee, and it reacts. And as part of that part of your body's fight or flight response is that your immune system revs up. It says, hold on, we might get hurt here, so let's start wound healing, let's start reacting to the fact that we might get hurt. So we have a biological explanation and a practical explanation for why we're seeing this immune system response to an adverse social exposure. Why does this become important? Because immune system dysregulation is important in a lot of important high-burden chronic diseases or processes involved in these chronic diseases. So when we look at cancer, it's involved with cell proliferation, with resistance to apoptosis, with genomic instability. We can look at atherosclerosis and see that it's associated with plaque formation and plaque rupture. We can look at diabetes and see that it's associated with insulin resistance and beta cell dysfunction. So you can stress me out. If I get stressed out enough, it has this whole biological cascade that then puts me at risk for a lot of different types of diseases. So I wanted to ask a question. I do some of this work previously. I said, well, okay, I've seen this study look at this exposure and gene expression, this exposure and gene expression. What if we look at all of the same people, try to look at a bunch of different exposures? Can we see some exposures hit and some exposures not? And then which exposures might be rising to the top? I used data from a study called MESA, it's the NHLBI or NIH, National Heart, Lung, and Blood Institute study from NIH. And the utility of using MESA is that they had info on both social and genetic factors. So typically in these studies, you have really good genetics data or really good social data. MESA was unique in that it had both, so it allowed me to ask about a range of social environmental exposures and gene expression. So I looked at 6 different exposures. I'll kind of go through them a little quickly. One looking at socioeconomic status in adulthood and childhood, a measure of perceived stress. These were assessed via survey, so people filled them out, and it's one of those surveys that was Likert scale, so it was like always, sometimes, usually never. I looked at loneliness. I used the same measure of loneliness that Steve Cole used, as I mentioned in the paper earlier. Major or lifetime discrimination, so that's how you've ever been discriminated against in one of these fields. Then I looked at chronic burden. So chronic burden is a measure of chronic stress. So it's stressors that they said that they had experienced for at least 6 months or more, and they considered it to be moderately or very stressful. And then lastly, to throw in something positive, I looked at social support to try to see if social support as a positive exposure would have an influence. I looked at 4 biological processes: chronic inflammation, inflammatory response, regulation of the inflammatory response, and the immune response. And what I found was that for chronic inflammation, I found an association for loneliness, chronic burden, and discrimination as being associated with inflammation-related genes. And then additionally, for discrimination as an exposure, I found that it was associated with gene expression and also in the inflammatory response, immune response, and the regulation of the inflammatory response. Now, how might this relate to cancer specifically? So I draw in this paper where researchers were looking at social isolation and whether it was associated with cancer-related gene expression patterns in breast tumors. So we know that social isolation activates the body's fight or flight response. One of the things of fight or flight response is a change in the tumor microenvironment, specifically what's known as an epithelial to mesenchymal transition and having an upregulation of what's known as M2 macrophages. So it's very specific to cancer. And they wanted to know, is there a difference in expression of those patterns by people who say that they're socially isolated? And they found that people who said they were socially isolated had upregulation of these specific genes. And these are amongst people who had cancer. And then on a positive, I guess, a potential intervention, these other researchers, they wanted to know if an increase in mindfulness-related well-being can affect CTRA, conserved transcription response to adversity, gene expression in breast cancer survivors. So they actually tried to do a 6-week intervention to try to see if a mindfulness intervention increased well-being. And for those people where the mindfulness intervention increased well-being, they found a change in their gene expression as well. So it may be interventionable that we can affect our gene expressions that are in regards to these social factors. And then there's the question of can epigenetics play a role? So, typically we used to think about genes as on-off switches, that you were born with the variant, you had the variant throughout your whole life, and that was just it. More recently, we're starting to investigate this idea of epigenetic modifications. And essentially, they're things on your genes that are kind of like dimmer switches. And they can be environmentally influenced and control how much or how little of a gene that you start to make. So that's just a picture of kind of what they look like. So I've worked on this paper, I was co-author on this paper that asks, is smoking associated with DNA methylation in African Americans? So this was in a study called Intergen, it's a study of African American women in Connecticut, and then reproduced in a study called Genoa, which we also used African Americans. And we wanted to know if smoking is associated with DNA methylation. So this is known as a Manhattan plot. Essentially, if there is a dot above that line, that means that the association is statistically significant. And what we found is that there were 26 methylation sites for where we could find an association between smoking with the methylation site. So we're starting to uncover the how. So we know smoking is associated with cancer, but we're starting to try to get to the biological mechanism whereby this might be working. And of the 26 methylation sites that we found, we mapped them on and found that there were six of the associated genes were cancer-related genes. So we can start to say, okay, we know smoking is cancer. We know smoking is actually affecting the expression of these genes, and the expression of these genes are how we're starting to see cancer. Before I joined this lab group, they asked a question that I also thought was interesting, so I wish I got there a little bit sooner. But they said, is racial discrimination associated with DNA methylation in this same group? Same Manhattan plot, same kind of Manhattan plot idea. Above the red line means that it was statistically significant. And they found that there were 9 methylation sites, where they found that racial discrimination, being exposed to racial discrimination, was associated with DNA methylation. So again, whereas like my other study said that, okay, we see that DNA, we know that discrimination is associated with health and disease, we're starting to see a how. We're starting to see this actually affecting the expression of our genes. So if we take a step up, I was trying to bridge between genes and our environmental exposures, but we know it's not just our environmental exposures that matter. So if I draw back again on this diagram, is that we need to actually take a step up and then look at, well, what happens by neighborhood level factors. So one of the leaders in studying neighborhoods and especially racial inequities is David Williams from Harvard. And he argues that racial residential segregation is a fundamental cause of racial health inequities. So let me just talk about it like this way. So a fundamental cause involves access to resources that helps people avoid disease and their negative consequences. So for example, so what he's saying is that if you geographically separate people by race, allocate resources to one neighborhood differentially from the other neighborhood, that's the fundamental reason that we see the racial health inequities that populate every single study of racial health inequity that we ever talk about. And in general, these flexible resources are things like access to healthy foods, opportunities for physical activity, it's medical care, different types of treatments and screening, quality of education, and things like that. So this, I'm highlighting a review article that looks specifically at neighborhood segregation and cancer racial health disparities, and they say that segregation contributed significantly to cancer and racial cancer disparities in 70% of analyses. So they looked across 17 different studies, created a summary of those 17 different studies, and said that even after controlling for socioeconomic status and health insurance, that segregation was a leading reason why we see disparities. And residing in a segregated neighborhood was associated with higher odds of late-stage diagnosis of breast and lung cancers, mortality rates, and lower survival rates from breast and lung cancers, as well as higher cumulative cancer risk associated with exposure to ambient air toxins. And acknowledging that I am in Iowa right now, I also wanted to highlight this paper that looks specifically at neighborhood deprivation and see if that was associated with cancer screening. And this was a study in the Midwest. So they combined information from Minnesota, Iowa, and Wisconsin and created a measure of neighborhood deprivation. So it was a measure of 17 different census tract variables, things like median household income, poverty level, level of education, and tried to see if looking at neighborhood deprivation was associated with who was getting screened for cancer. And what they found is when they looked at breast cancer, they looked at cervical cancer, they looked at colorectal cancer, and for each type of screening, the more deprived the neighborhood, the less likely they were to be screened for cancer. And probably one of my favorite studies is by a guy named Tom Levis. He did his work while he was at Hopkins. He's now dean at Tulane. And he wanted to know what would health inequities look like in racially and socioeconomically integrated communities. So if you put black people and white people in the same neighborhoods under pretty much the same socioeconomic conditions, would we still see all of the inequities that every epidemiological study says that we have? And the answer was no, not really. So looking across different studies, they were either eliminated or severely attenuated if you put people under the same environmental conditions. We talk about racial segregation, but racial segregation is not a natural phenomenon. It's not like the country started because black people said, we want to live over here, and white people said, we want to live over here, right? It was really by design, it was by policies. If we take another step up on my ladder here, that created the racial segregation that we see. Here I borrow some information from the FHA manual of 1930. These might look a little bit familiar, but this is DC's redlining map, where the FHA would literally go out and grade the neighborhoods. And the way that they graded it, most of them are graded on A to D. I'm from the DC area. Ours happens to be on an A through H scale. And they describe A neighborhoods as the best residential area and are occupied chiefly by persons in the highest income groups, whereas H neighborhoods are more for the result of the exploitation of the Negro rather than a planned development, literally written into federal housing guidance on how to insure mortgages in the 1930s. Further, it said, if a neighborhood is to retain stability, it is necessary that property shall continue to be occupied by the same social and racial classes. In communities where there is any possibility of Negro infiltration, application for the insurance should be rejected. So as it kind of makes sense as a consequence, less than 2% of the approximately $120 billion of housing financed by the Federal Housing Administration and the Veterans Administration between 1934 and 1962 went to non-white residents. So this just starts to get to looking all the way up to policies and why we're seeing the inequities that we see more downstream. Now, we could say, okay, that was the 1930s, right? It's 2022. One of the things that came out of civil rights legislation was the Home Mortgage Disclosure Act, which required financial institutions to maintain, report, and publicly disclose loan-level information about mortgages. So there's a database, publicly available database, available. And what researchers in recent years, past 5 years or so, have started to do was use this database to try to identify areas where there's, quote, contemporary redlining. So where are the areas in the country where black people still can't get mortgages at the rate that white people can after accounting for economic factors? So that's the way that scientists are currently measuring contemporary measures of redlining. And then what they're finding is that living in a red line neighborhood, whether you measure it by these 1930s maps of what was a desirable neighborhood and what was not desirable, or whether you measure it by contemporary measures, measuring who's getting mortgages where now in 2022, it's been associated with late-stage cancer diagnosis and mortality. So this is the way in which we're seeing policy now also affecting our health inequities. So if I think about everything that I said today, it's really been to drive home the point that we need to think about health at multiple levels, that we could think about it at genes, we need to think about it at our individual level stressors, we have neighborhood level factors that we know that are contributing to it, we have policy level factors that we know that are contributing to it, and that if we're ever really going to get to precision health, precision medicine, that really we have to think about it at all of these levels. We have to work together. We have to be dually trained and think about the bigger picture at all times. So what's just one small way that we can do this? Like many epidemiologists, when COVID hit, I turned my work a little bit towards doing COVID-related work. And one of the activities that I'm involved in now is known as a learning community. So essentially what we try to do, or we try to understand that trying to get people vaccinated was not really just about the individual person's decision, that it was really about the environment and the culture in which they lived, and we needed some sort of concerted response that wasn't just a one-on-one conversation, but that got to people or efforts at the community level, at the local level, at the state level, at the national level, and we needed everybody working together to try to distribute this healthcare resource. So we have partners from national organizations, we have partners at pharmacists, we have community-based organizations, and everybody working together for this one thing. And what we do in the learning community is that we have a virtual community where people in California, people in Michigan, people across the nation, it's about 500 organizations that we link together so that we can try to get them information correctly, that we have a members-only website so that they can interact with each other. So if I want to say who else is studying Hispanics in New Mexico, I can find somebody else in the directory who is also studying that. We have communities of practice groups to say, if you focus on this population, let's link you with others who focus on this population, thinking the idea that we're just better together, right? Some organizations have 3 people running the show, some people have 10, but if we can use our strengths together, if we can share our resources, that we'd be better at all of this. And I say that to say, I know that's completely about vaccines, but I think that structure is applicable to any kind of disease that we go after. So can we do this with cancer? Can we link together our physicians and our researchers and link them with the on-the-ground community health workers? Can we link them with our urban planners who are designing the neighborhoods in which we live? Can we link them with our policymakers? And can we use that kind of structure to collaborate and work together? And my hope is that we can do that, and that would be a successful approach to addressing cancer, addressing cardiovascular disease, addressing diabetes, addressing them all together and just improving overall health. And thank you so much for your attention. Thank you for hanging out this late.